So you see, my topic is obesity modulates the gut microbiome in triple-negative breast cancer. So I thought in 10 minutes, it's not possible to showcase my work because it will take some time. But what I am going to show you, I'll walk you through some of the coming treatment revolution that is happening in this field. That is very interesting, what's happening. I am just playing a part along with Lucio Mille, Dr. Mille and Dr. Hussain is here. They are also a big part of our team. And we are actually trying to, see what's happening here. So I am an assistant professor in the Department of Genetics, LSU Health Science Center. So my previous speaker discussed at length. So, the triple-negative breast cancer, although it accounts for only 15%, 12 to 15% of all the BCs, it is the most aggressive form, largely due to the fact that it is presented at a higher grade, particularly diagnosis, and then bigger metastatic potential. It's a higher rate of resistance and relapse, and also very, very, dire treatment outcome. That's why it is so breast cancer as a whole, the mortality rate is second highest, but triple-negative breast cancer contributes to the highest degree here. And also it is disproportionately affects the African-American population, particularly in the southern United States. It's very prevalent. And also we can see this disparity. We don't know the causes of this disparity well, but that is what is happening right now. People is trying to dissect what's happening in the African-American population. However, in the obesity and overweight population is huge, particularly in the southern state. However, triple-negative breast cancer is a heterogeneous disease. And in this landmark paper in 2011, what they have shown by molecular subtyping, which is still the best way to characterize, cancer on the basis of DNA gene expression profiling. So they actually showed that triple-negative breast cancer, even though it is 12%, but still there are 6 different subtypes of triple-negative breast cancer. Basal-like 1, 2, immunomodulatory, mesenchymal, then followed by mesenchymal stem cell, and then androgen receptor signaling. And however, the 75% of the triple-negative breast cancer is the basal-like subtype, and it tends to be, less differentiated with the worst prognosis. Now, I'm just talking about the treatment, so there is no, as everybody here, that there is no receptor expression status or very low. So basically you cannot target the triple-negative breast cancer. So chemotherapy continues to remain the mainstay of treatment. However, in the last few years, I've witnessed some revolution, particularly some targeted drugs, particularly the BRCA1 mutation. If you have a germline BRCA1 mutation that happens in 10% of the TNBC, you can target them with the PARP1 inhibitors. They are pretty good about that. And also in the very recently, in the last 2-3 years, the interfering with the PD-1, PD-L1 pathway has revolutionized the cancer treatment. So breast cancer is also one example, particularly in the TNBC. In a lot of the TNBC tumors, they are little immunogenic compared to the ER-positive background. So they have a subgroup. Again, it's a subgroup of TNBC. They express PD-L1 in their immune cell population. So that could be, modulated with the immunotherapy, and they have actually FDA has approved. The first wave was PD-1 and anti-PD-1 monotherapy. It was good. It was efficacious, and the second wave they actually tried with immunotherapy with the existing chemotherapy as the first line treatment in the metastatic TNBC or the locally recurrent TNBC. And it was approved by the FDA because they have a statistically significant, progression-free survival and overall survival with this combination treatment compared to chemotherapy alone background. And later, they also approved in the first-line treatment in the neoadjuvant setting in the very early TNBC, the same treatment group approved. So here, basically anti-PD-1 therapy, they approved the anti-PD-L1 also, but somehow initially FDA approved, but they later withdrew. for the anti-PDL1 because the progression-free survival rate, it did not actually match with the previous estimates. So right now, the anti-PD1 or pembrolizumab, which is the most efficient one or the most used one, is commonly used in the immunotherapy background. And now they are trying to modulate this immunotherapy, refine the immunotherapy with that other targeted therapy. That is actually a third wave is going on. So with this in background, so overarching challenge remains, the response to immunotherapy remains suboptimal. This is largely due to the fact that with the use of the immunotherapy, probably the TNBC at the first instance, they respond to immunotherapy better because they have immunogenic potential, apparently, from the ER positive. However, when you actually treat them with immunotherapy, first of all, there is adverse immune-related event. They also occur pretty much in this patient, and also durability actually subsides from time to time because again the exhaustion of the PDL1 and the PD1, which was the very first case due to the presence of it, you actually apply the immunotherapy, but later it actually diminished. So the duration is a big challenge. So obesity and cancer, my previous speaker actually mentioned a bit about it. So according to the US Center for Disease Control, you know, the cancers associated with overweight and obesity is a thyroid, liver, kidney, and ovarian cancer. And in 2005 to 2014, disparities between sexes is also very concerning because there is 55% of the women diagnosed with cancer. These are overweight and obesity related compared with only 24% in the men. So it is tough, given it is most concerning because women, the chance they actually more often than not, they actually resist smoking, and alcohol addiction is less in the women. Most of the disparities due to endometrial, ovarian, and postmenopausal breast cancer, which Dr. Shu mentioned, which together makes up around 42% of the cases of obesity-related cancer. And about 2/3 of the adults in the US are considered overweight. It is going to be 13% in 2035. And overweight and obesity-related cancers also differ downward trend seen with other cancers, growing by 7%, contrasted with the 13% in most other cancers. That is also one of the red flag. So obesity, Dr. Shu mentioned again, yes, there is not a very consensus here, but however, if you take the TNBC population, and particularly it afflicts the African American population disproportionately, there is a lot of obesity. And in the retrospective study in 640 TNBC patients in West Virginia, there is 5% normal TNBC patients. And another independent study confirmed that obese women have 20% higher incidence of TNBC compared to normal individuals. So yes, there is a controversy, but there is, of course, some documented examples that obesity and TNBC is intertwined. Now, one of the fundamental mechanisms that what we are witnessing, it is a kind of a revolution going on, is and also it is in the NCI's priorities is to modulate because obesity is a low-grade inflammation, as Dr. Shu pointed out and everybody knows. There is a lot of inflammatory changes happening in the body. So if you can actually control these inflammatory changes, then you can do, and obesity, one of the important reasons that was actually proved back in 2005, that was the first paper in PNS, where they proved that in the obese mice, the gut microbiome composition and function changes drastically from the lean mice. That was the first paper that actually, created the whole gamut of opportunities. And later, in the last couple of years, if you see the obesity-related and gut microbiome-related papers published is skyrocketed, even including all the high-flying journals. Even if you see interesting how obesity can modulate. So just to give you an example, in the Japan, the incidence of breast cancer as such is very, very low. But however, when the same Japanese patient, normal Japanese comes to USA as an immigrant in 30 years, she actually attains the chance of having a breast cancer in the Japanese, normal Japanese patient is the same as in the United States. So in 30 years, probably his gut microbiome changes, and there is a same paper in my lab which shows US immigration westernizes the human gut microbiome. It is very interesting, insightful. And even in the last month, even this month, 15th September, it happens, the microbiota in Cell paper, another, microbiota imbalance induced by dietary sugar disruption, immune-mediated protection from metabolic syndrome. That is a huge paper that came. And now, what is the best chance without putting the patient through adverse effects? The best chance is the gut microbiome. So there are hundreds of companies, there is synthetic biology, there is chemical engineers who are working on it. It is just huge. And the probiotics are designed to improve your microbiome because it is well-known fact that it actually affects the health and benefit. But however, I don't think that one shoe fits all. It is going to work for every patient because it could be what is a TNBC patient, maybe a change in the microbiota, particularly one or two species, is all that it matters compared to the other individuals. So that is going to take a long, long time. In colon cancer, of course, microbiota, I'm coming there again, but in other cancers, it is, I don't know whether it is causative, but whatever the signs, the documented evidence is showing, it is definitely contributed. In colon cancer, in the set of colon cancer, it is causative. But in lactobacillus and bifidobacterium have been used in yogurt pills, probiotic powders for decades. It is showing the results. Now, in our paper, which Lucio and Dr. Hoshen and Dr. Mille is also part of this in big time, what we have shown also, the obesity alters the gut microbiome. So in my lab, if you see the NMDS ordination plot, which actually shows the group similarity or dissimilarity to the others. So we have actually, we induced high fat induced dust in FBP background mice. So there was lean tumor, there was lean mice, there was obese mice one group, and another lean, obese tumor and lean tumor. So you can see there is a distinct ordination plot. The groupings is very, very different. And also, if you see the lean tumor and the obese no tumor, the shifted away from each other. There is a nice groupings here. And also, if you see the tumor versus no tumor in the obesity tumor, here it is tumor and obese and lean. In the obesity and the lean tumor, there is a much higher, what is this called? The diversity index, so alpha index. So the diversity index, and we have seen that the alpha diversity and the beta diversity is both higher if it is the lean and the obese tumor. And we have seen not only the obesity, but obesity tumor interaction also imparts some kind of dysbiosis. But whatever our biostatistician told, that obesity imparts the biggest effect on the statistical significance because our sample size was not big enough to tell you exactly how the tumor obesity interaction actually plays a big part. But the obesity, beyond any doubt, it imparts a very big part. And also here we can show that the changes in obesity, also you can see the species differences. We have seen the Akkermansia is actually, associated with a big increase in the species population, almost 70-80% when it is obese versus the control tumors. So now, sorry. So now there is a big how to actually modulate obesity hard data, because science is not about hype and hoopla. It is hype because you need to prove it by hard data. So it is. Can you hear me? Okay. So now in this 2017, in the same issue in Science, it is the same issue. So 3 independent groups, they approved. So in melanoma patients with more gut microbiome diversity respond better to anti-PD-1 therapy. And in the same issue, another group documented that gut bacteria influence response in epithelial tumors. They have shown bacterium is associated with the subset of colorectal cancer specifically. So that's why to test your gut microbiome. That is the FDA approved, and this Fusobacterium nucleatum is associated with the subset of colon cancer. And if you treat that cancer patient, colorectal cancer patient, with the antibiotics, they will respond better to the PD-1 or anti-PD-1 therapy. So this is actually what's brewing in. So what we have seen, it is definitely rekindled hope. And in the new avenues, basically obese patients also, what we have seen, this is again in the clinical trial. They respond better to immunotherapy in few cancers, including melanoma. But again, it is not durable, and it associated with immune-related adverse events. So modulating gut microbiome to enhance the efficacy of immunotherapy in TNBC obese patients, nobody has ever seen. We are trying to do it, definitely. We are awaiting for the grant, but we have proved that obese individuals, they have a different microbiome profile. So we will actually in the 0321, that is claudin low tumors. But if you can actually see some, gut microbiome can enhance the efficacy of immunotherapy in the TNBC obese patients, that is a far, but at least first in mouse, that will be a big thing to cross. And then, so this is a new avenues. There's a lot of stuff going on. This is still in the very early stage. A lot of it is not a consensus. Some cancers are showing promise, some cancers are not showing promise, and some are telling exactly the opposite. So it is still, the dust is not settled yet. But definitely, the sky is the limit. This is all new thing. Thank you. And again, thanks, Lucio and Fokul, for your contribution in this one.
Thank you so much for this excellent overview of the relationship between microbiome and triple-negative breast cancer. Are there any questions?
Yeah, very interesting talk. I see the obese and non-obese, between the obese and non-obese, the microbiome difference is not as huge as in obese population in mice with tumor, without tumor, it seems to me, according to your figure. Is that true?
You know what, Dr. Shu, the whole problem is we published this paper in a decent journal. To be frank, it was beyond our expectation that it would have been expected in the first shot, because the sample size was so low. It was only 5 group. So the biostatistician from the company who actually did our work, they told anything that is very important, significant, we can tell beyond any reasonable doubt, it is the obesity. However, beyond that, we cannot tell that the tumor, how far the tumor plays a part, how far tumor obesity interaction in the interface plays a part, we cannot tell. But obesity definitely plays a part that is significant, because sample size is so low, and particularly, as you know, in the when you are doing the microbiome modulation, the changes, is confounding factors are so much, even cage to cage, even from room to room, even from center to center, it is so huge. You need to put at least 20, 30, 40 mice in order to do this kind of experiment. But this kind of resource, without a big grant, is not possible. But yes, you are right. So anything I can tell with a grain of salt. But having said that, it is published. We are surprised, but it was published in decent journal.
Another question is related to the, how those microbiome may regulate tumor progression and immunotherapy. There are many studies have shown the metabolite, produced by those microbiome is quite different. Have you thinking about exploring that direction?
Yes. Basically, what we are proposing, I am actually submitting a DoD grant there exactly because it has been shown that short chain fatty acids, particularly the butyrate, pyruvate, because when you are from lean to obese mice, it has been consistently shown there is a 50%, almost in many studies, it is consistent. There is a 50% reduction in bacteriorides population, and there is a 50% increase in the Clostridium population. And that actually disturbs the production of short-chain fatty acids. And if you disturb the production of short-chain fatty acid, it disturbs particularly in breast cancer perspective, the estrobolome, basically the enteric bacteria that degrade the estrogen and everything. So basically you are disturbing the whole estrogen signaling pathway and everything. But definitely that is something, if you do the targeted metabolomics, what are the fates of the short-chain fatty acids? They can give you some indication what's happening. Particularly in my aim, we are proposing the TNBC patients also. So we are going to do from their plasma the short-chain fatty acids. So basically the TNBC patients pre and post anti-PD-1 therapy in obese TNBC patients. And we will collect the plasmas also. And we will do the targeted single-chain, short-chain fatty acids and see what are the response, what are the difference between the responders and the non-responders, if there is anything in addition to the metagenomics and shotgun sequencing.